Introduction to Enzymology. Let's start at the beginning. Enzymology is the branch of biochemistry that deals with the study of enzymes, the biological catalysts that make life's chemical reactions happen at rates fast enough to sustain life. Think about it this way: if you were to leave a lump of sugar in a cup of water, it would eventually dissolve on its own. But if you stir it, the process speeds up. Enzymes are like the ultimate stirrers. They accelerate chemical reactions, sometimes by factors of 1,000,000 or more, and they do it under the mild, watery, body-friendly conditions of living cells. Enzymes are usually proteins, though a small group known as ribozymes are made of RNA. Whether protein or RNA, they all share the same role: to lower the energy barrier for reactions, making them happen more quickly and efficiently. 2. Classification of enzymes. The International Union of Biochemistry and Molecular Biology (IUBMB) classifies enzymes into 6 major classes based on the type of reaction they catalyze. Let's walk through them, one by one. 2.1 Oxidoreductases. Function: catalyze oxidation-reduction reactions, where electrons are transferred from one molecule, the donor, to another, the acceptor. Example: lactate dehydrogenase, which converts lactate to pyruvate during cellular respiration. 2.2 Transferases. Function: transfer functional groups, such as metal or phosphate groups, from one molecule to another. Example: alanine transaminase, which transfers an amino group from alanine to alpha-ketoglutarate. 2.3 Hydrolases. Function: catalyze the hydrolysis of bonds, breaking molecules apart with water. Example: amylase, which breaks down starch into sugars. 2.4 Lyases. Function: break bonds without using water or oxidation, often forming double bonds or new ring structures. Example: fumarase, which converts fumarate to malate in the citric acid cycle. 2.5 Isomerases. Function: rearrange the structure of a molecule without changing its composition. Example: phosphoglucose isomerase, which converts glucose 6-phosphate to fructose 6-phosphate. 2.6 Ligases. Function: join two molecules together, usually with the input of energy from ATP. Example: DNA ligase, which joins DNA fragments during replication and repair. 3. Structure and active sites. Enzymes are not just random chains of amino acids. They are folded into very specific three-dimensional shapes that allow them to do their job. At the heart of each enzyme's function is the active site, a special region where the substrate, the molecule to be acted upon, binds. The active site is shaped in such a way that it recognizes and interacts with the substrate through various weak forces, hydrogen bonds, ionic interactions, and hydrophobic effects. Think of the active site like a glove designed for a specific hand. The substrate fits into it perfectly, a concept often summarized as the lock and key model. But real enzymes are a bit more flexible, often adjusting slightly to fit the substrate more snugly. This is known as the induced fit model. 4. Mechanism of enzyme action. Enzymes work by lowering the activation energy, the energy needed to get a chemical reaction started. Without an enzyme, reactants might need to collide with enough energy and in the right orientation for the reaction to occur. With an enzyme, the active site stabilizes the transition state, making it much easier for the reaction to happen. The general steps are: substrate binding, the substrate enters the active site. Formation of enzyme-substrate complex, temporary bonds and interactions stabilize the arrangement. Catalysis, the enzyme facilitates the transformation of substrate into product. Product release, the newly formed product leaves the active site. Enzyme recovery, the enzyme returns to its original state, ready for another cycle. 5. Factors affecting enzyme activity. Enzyme activity is sensitive to several factors. Let's explore them. 5.1 Substrate concentration. At low substrate concentrations, enzyme activity increases rapidly because there are plenty of active sites available. As substrate concentration increases further, the enzyme becomes saturated, and activity levels off, the enzyme is working at maximum capacity. 5.2 Enzyme concentration. If you increase enzyme concentration while keeping substrate constant, the rate of reaction increases proportionally. More enzymes mean more active sites working at the same time. 5.3 Temperature. Temperature affects enzyme activity in two ways. Increase. Higher temperatures generally speed up reactions due to more molecular collisions. Too high, enzymes, being proteins, can denature at high temperatures, losing their shape and therefore their activity. 5.4 pH. Each enzyme has an optimal pH at which it works best. Deviations from this can alter the charge and shape of the enzyme and substrate, reducing activity. 5.5 Inhibitors. Inhibitors slow down or stop enzyme activity. Competitive inhibitors compete with the substrate for the active site. Non-competitive inhibitors bind elsewhere on the enzyme, changing its shape and reducing activity. 6. Michaelis-Menten concept. While we won't use the full equation here, the Michaelis-Menten model is important because it describes how enzyme activity changes with substrate concentration. It introduces two key ideas. Vmax, the maximum reaction rate. Km, the substrate concentration at which the reaction rate is half of Vmax, a measure of the enzyme's affinity for its substrate. A low Km means the enzyme binds tightly to the substrate, while a high Km means it binds more loosely. 7. Cofactors and coenzymes. Many enzymes require extra non-protein components to work. Cofactors, usually inorganic ions like magnesium or zinc. Coenzymes, organic molecules, often derived from vitamins, such as NAD+ or coenzyme A. Prosthetic groups, tightly bound cofactors that remain attached to the enzyme. Without these helpers, some enzymes cannot function. 8. Enzyme specificity. Enzymes are remarkably selective. 8.1 Group specificity. An enzyme might act on molecules with a certain type of bond or functional group. Example, pepsin cleaves peptide bonds involving aromatic amino acids. 8.2 Optical specificity. Some enzymes distinguish between different stereoisomers. Example, L-amino acid oxidase acts only on L-amino acids, not deforms. 8.3 Geometrical specificity. An enzyme might act only on molecules with a specific shape or arrangement of atoms. 9. Regulation of enzyme activity. Cells regulate enzymes to control metabolism. Allosteric regulation, a molecule binds at a site other than the active site, changing the enzyme's activity. Feedback inhibition, the end product of a pathway inhibits an earlier enzyme, preventing waste. Covalent modification, enzymes can be activated or deactivated by adding or removing chemical groups, such as phosphates. 10. Applications of enzymes. Enzymes are not just important in living organisms, they are widely used in industry and medicine. Industrial, brewing, cheesemaking, textile processing, detergents. Medical, diagnostic tests e.g., glucose oxidase for blood sugar monitoring, drug manufacture. Research, molecular biology tools like DNA polymerases and restriction enzymes. That completes Module 1 Enzymology. We've covered what enzymes are, how they are classified, how they work, what affects their activity, how they are regulated, and where they are applied.